And gotta make sure this is open. Okay, awesome. All right, but first we're going to take just a little peek at this chapter 18. Okay, maybe. There we go. Okay. And the big thing about chapter 18 is there is a lot of footnote text under there. It doesn't show up in the slides, but I know there's a couple slides where that is actually pretty helpful. So we've talked about fatty acid oxidation. And in previous chapters, we talked about glucose, using that for energy. So glucose, fatty acids used for energy. Now we're going to look at protein used for energy. And I think about this because I come in here and my breakfast is often beef jerky. Why? Because it's protein. Okay. But amino acid oxidation and the production of urea are intertwined. So we're going to talk about the urea cycle a little bit as well too. Figure out how proteins are digested, how urea is made and excreted, and our energy from protein. It's very interesting because my 6-year-old daughter, who I think because my son is studying physics in his middle school math now, wants to be included. So she's talking about how she wants to study math and science and wants more homework. And tomorrow she wants to go to college. I'm like, let's slow down just a little bit. When we cook supper, she will identify the meat and say, what muscle is this? What muscle does this come from? So, and then that's going to make muscle. Does this muscle make a particular muscle here or something like that? So, it's something that, we do on a daily basis. As long as we're eating food, we are, dealing with all this biochemistry that we're talking about here. And it's understandable. It's just the depth we're going in starts to, get a little intertwined. The connections can be a little confusing at times. So this chapter is really kind of where we start, where I start slowing down. But organisms will use amino acids as fuel. This varies from organism to organism. We talked about hibernating bears yesterday, or last class, and how they're going to primarily be using lipids during that hibernation period. But if we look at bears being omnivores, if we look at a carnivore, almost all of their energy needs can be met from crunching up another animal in there. Think of hyenas and whatnot. All right. So microorganisms will also scavenge amino acids from their environment when they need fuel. So we think of bacteria, we often grow it on some type of sugar solution, but if there's no sugar, then they will scavenge for amino acids. Herbivores use minimal amounts. So our cows out in the field there are not going to get a lot of energy from amino acids. All right. Now it's interesting because we tend to think of plants as they don't use them as fuel, but they will degrade them to make other metabolites as well too. So there's still these metabolic pathways in there. They're just not being shunted into energy production. That energy production is coming primarily from photosynthesis, but they will metabolize amino acids. Of course, we don't care about plants in this class, so we're going to ignore them for the most part. All right. So where do we get these amino acids? All right. A lot of them come from leftover amino acids from normal protein turnover. We talked about, and sometimes I think we don't emphasize it enough, but proteins in the body have a lifespan. Aside from a few ocular compounds that last for like decades, if you think of the liver, the turnover rate for proteins in the liver is about a day. So you're constantly synthesizing new proteins, which means you're degrading the old proteins. So you break them down into their component amino acids, and that's one source of amino acids. Also, I had my beef jerky this morning, so dietary amino acids there. Chewing on some dried cow. I don't know. Sounds terrible when you say it that way. And then the other thing that can happen is if we go into a starved state, we can break down proteins in the body to supply amino acids for energy. So we see this in starvation. You can also see this in some disease states, most common of which is diabetes mellitus. I think I mentioned, concentration camp survivors, emaciated, basically their bodies completely exhausted, fatty acids, no very minimal carbohydrates from the thin gruel that they were often served. So, they cannibalize their own muscle to get energy in there. All right. So if we start with our dietary proteins, the first thing that's going to happen when I chew up my beef jerky is it is going to be hydrolyzed by various amino acids. So we have these long protein strands. We're going to start cutting them up. And pepsin, trypsin, and chymotrypsin are the three major ones in the stomach and the intestine that will recognize particular sequences and then make a cut after those sequences. We also have aminopeptidase and carboxypeptidases, a couple of those that will degrade them in the small intestine as well too. But pepsin is usually kind of like the first one. So interesting thing, I'm sure you've probably heard, if you hold a piece of bread in your mouth for long enough, it'll turn to sugar, right? Because your enzymes will cleave the carbohydrates that are making up the bread and eventually cleave them into glucose. So you can start digesting carbohydrates directly in the mouth. If I hold a piece of beef jerky in my mouth, it's just going to be beef jerky. I have to actually get it into the gut for it to be digested in there. All right. So here we are in the stomach. Again, we see we have this lovely low pH, so that's going to help our pepsin work. Our pepsin is actually in an inactive form, pepsinogen, so that when we actually start to eat, that pepsinogen can be activated into the pepsin molecule and then start cleaving proteins. And then we have our stomach lining that will secrete a lot of this. Our gastric mucosa, our various like these chief cells. I think I should call them chef cells, right? Because but anyway, and all these organs are kind of closely interconnected in here and communicating. We'll activate proteases in here, our pancreatic duct. When you think of our islets of Langerhorn secreting our insulin, that's because once we get sugar in the gut, that's the response there, because these are close in space. They're communicating with each other. But in this case, for proteins, again, we're going to see the mucosal lining in here is going to be where we hold a lot of these pre-proteins or proproteins in there that can be activated. I always find it interesting too, the architecture of the cells of the mucosa in there. There's a lot of space in there, just like we saw in the mitochondria. We have the cristae, those fingers in there, so you can have lots and lots of electron transport chain activated at the same time. Here we have these little like fingers that kind of stick in there, so there's lots of spaces for the proteins or carbohydrates or lipids or whatever to interact with the mucosal cells in here. Also, that contributes to their turnover as well too, because there's so much exposure and the environment is very acidic. So we can start digesting all sorts of stuff in there, sometimes even our own stomachs. Okay. But anyway, all this process leads to, we have a whole bunch of these peptidases, trypsin, chymotrypsin, pepsin, that are all breaking down amino acids. We get them into the individual amino acid components in there. And then we have basically two things that can happen to them. We can recycle them into new proteins, right? So, like my daughter, we eat some chicken, breaks it down into component proteins. It goes to her muscles and makes new muscle proteins in there. Right. So we just kind of, I love the Lego analogy. You break down the Legos into the component little pieces, and you can build something new with it. All right. So, so that's what we're doing here, recycling the new proteins. That's one option. We'll talk more about that later on in this class, but not for exam 2 material. We're going to focus on the second option, where we can oxidize them for energy. And to do this, we're going to have to remove the amino group in there, and that's going to feed into the urea cycle. So we're going to talk about nitrogen balance. We want to maintain homeostasis of the concentrations of amino groups in there. So if we're eating lots of protein, we're getting lots of nitrogen from those amino groups in there. So we can feed that into the urea cycle and excrete that. Right? We can also see the other parts of the molecule going into central metabolism. And we've sort of hinted about this. Some of those can feed into anaplerotic reactions, feed into the glycolytic cycle, or feed into the citric acid cycle. Okay. All right, so we can see these kind of being connected here as well. So here's our amino acids. Our amino group goes over here. We can use that to synthesize new things like nucleotides or biological amines, or we can feed it into the urea cycle. And we're going to talk about carbamoyl phosphate a lot, carbamoyl phosphate synthetase. So CPS 1 and 2. I don't know about North Carolina or any of your particular home states, but growing up in West Virginia, nearby Pennsylvania, CPS stood for Child Protective Services. So I always think of this as being like the chaperones coming in and say, okay, we're gonna take care of what's going on here. But our carbamoyl phosphate is kind of our first step before we get into the urea cycle, and then we can basically take excess amines and excrete them as urea. Okay. We also see our carbon skeletons, these alpha keto acids, alpha ketoglutarate, can feed into the citric acid cycle, and we can make our oxaloacetate, or we can make glucose ultimately from that, from gluconeogenesis. But these two are connected by this aspartate or genosuccinate shunt of the citric acid cycle. So if you take a look at that citric acid cycle, our succinate dehydrogenase, I guess somewhere at about, that'd be 7 or 8 o'clock in the citric acid cycle, there's a connection between that section and the urea cycle that we'll see there. Okay. All right. So let's start by talking about nitrogen. All right. And if you think of our atmosphere, we're talking, close to 80% nitrogen. It used to be much more than that until we start making oxygen there. But plants will conserve almost all the nitrogen. This is where we start looking at environmental impact. If we get rid of a lot of plant life, what are we doing to the nitrogen balance in there? But a lot of aquatic vertebrates release ammonia to their environment. So if you think of like swamp gas, it's always kind of thing I heard about, but I never really lived near swamps or whatnot. But a lot of these aquatic vertebrates will release ammonia. That's basically their excretion system. So it tends to be a passive diffusion from their epithelial cells, and the ammonia just kind of goes out into the water for release. So if you ever keep fish, that's the thing you have to keep track of, is like ammonia balance in the water in there, because they excrete ammonia. And then you wouldn't want to pour like bathroom cleaner in your fish tank. Anyway, most terrestrial vertebrates and sharks will excrete nitrogen in the form of urea. It's far less toxic than ammonia. All right. And it has a very high solubility, which is why our kidneys work. So we can dissolve the urea in water and then excrete it out as urine in there. If you look at birds and reptiles, they excrete nitrogen as uric acid, which tends to be insoluble and makes kind of a paste in there. This becomes important if you look at, think of lizards. A lot of lizards live in tropical or desert areas, so they need to conserve as much water as they can. So they kind of excrete this paste. If you've ever looked at bird poop on your car, you got that white sticky portion. There's our uric acid in there. Okay, and then of course humans and great apes. We're primarily going to talk about urea, but for solid waste we will see uric acid as well too. So looking at our forms of nitrogen in here, ammonia is the simplest one. We just, release ammonia. But again, you think of ammonia, it's like your kitchen floor cleaner or something, or bathroom cleaner. Just don't mix the ammonia and the chlorine bleach, because then you get chlorine gas in there, hypochloric acid, something like that. But anyway, ammonia tends to be rather toxic. So, if you are in a large environment or you have a constant influx of water in there, then that ammonia can kind of get washed away and it doesn't matter. But in an enclosed environment like a fish tank, then you can see a buildup of ammonia that can become toxic to the organisms in there. For urea, this is slightly different and far less toxic. Here we have two nitrogen groups around a central carbonyl carbon in here. That's our urea. And then our uric acid almost kind of looks like a purine in there, because we have our dual ring structure in here. Okay. And of course, you can probably guess that this does not look terribly soluble, whereas both of these are very soluble. All right. So what happens for organisms like us? we do not want to release free ammonia, because if we start releasing free ammonia, say in the bloodstream or in the liver or kidneys or wherever, we start seeing an elevated concentration of ammonia that rapidly becomes toxic. So instead, what we're going to do is we're going to transfer our nitrogen groups in here. So our amino groups are going to be, kind of shunted from one molecule to another by a series of transaminations. All right. So our transaminations are going to allow us to, find some common metabolites that then we can filter out into different, probably filter is the bad word there, that we can then shunt into different directions in there. So alpha ketoglutarate kind of sort of tends to be one of our central ones. Glutamate is also very important because if we look at this, if we deaminate glutamate, we can make alpha ketoglutarate and ammonia. Okay, if we add an ammonia to alpha ketoglutarate, then we make our glutamate. Okay, we can also add another amino group to glutamate to make glutamine. So our carboxylic acid becomes this carbamine down here. And we can remove that amino group, a single amino group from glutamine to make glutamate. All right. And in both of these cases, what we're seeing here is we're kind of transferring this amino group into the urea acid cycle to make urea. Okay. Our glutamine will often come from muscle and other tissues. Glutamine actually tends to be an internal storage mechanism for excess nitrogen. So if I gulp down a whole bag of beef jerky, it's probably a really bad idea. Then I'm going to actually end up with a lot of glutamine in my system that's going to be filtering into the liver so that I can start making a lot of uric acid and urea from that. Okay. If we start looking at muscle, alanine, we kind of talked about the actually, did we talk about the Cori cycle? I think we showed the Cori cycle. We're going to talk more about that today. But our alanine and lactic acid from the muscle, we can see that come in here. And again, with these alpha keto acids, we can combine that with alanine to make glutamate, and then again deaminate that to give us alpha ketoglutarate. And then of course what we eat, kind of feeding in here as well too. So the whole idea here is we got to, take care to maintain an appropriate concentration of nitrogen within the body. What was I going to say? Oh, yes. Okay. So as I drive into school, sometimes I listen to one of the comedy channels on the radio, and there's a guy today talking about, being a reluctant omnivore. He's trying to be a vegetarian because it's healthier and all that. And one of the things he was talking about was, it's like you never see a vegetarian go, oh, too many lentils, I think that. But lots of people will, bacon wrapped a Little Caesars pizza, that's too much. Yeah. And so you kind of get that bad feeling in there. So maybe by consuming a lot of proteins, I guess lentils will be protein too. Never mind. That's trying to go down there too. Anyway, I was reaching for something, but it's not there. Animal protein, plant proteins, mostly fairly similar. There are some differences. Okay. Anyway, we have a lot of proteins in the gut. We need to transfer these amino acids in here. So let's talk about how this happens. All right. And this, so far we've had, glycolysis and gluconeogenesis, fairly straightforward pathway. It's just one direction or the other. We just have a few roundabouts for the sticking points. Citric acid cycle, nice big curve in there. Fatty acid oxidation, we're just breaking stuff down. Well, here we need to break stuff down and take care of waste metabolic products in here, or recycle or something like that. So it gets a little dicier. So when we talk about aminotransferases, many, if not most of our aminotransferases are going to use pyridoxal phosphate as a cofactor. So pyridoxal phosphate is a mouthful. We're going to call this PLP for short. All right. So we're often going to see alpha ketoglutarate is going to be the molecule that accepts amino groups. Now, glutamate can sort of fill that role as well too, from glutamine and other places in there to make glutamine, but alpha ketoglutarate is going to be kind of the central one. And typically we're going to transfer one amine group to alpha ketoglutarate to give us glutamate. we can do a second one to get glutamine in there. Okay, and I mentioned L-glutamine acts as temporary storage of nitrogen in the body. If you have a lot of protein byproducts in there, then you'll see elevated levels of glutamine until they can donate that to either the urea cycle or for amino acid biosynthesis or other things in here. Okay, so this is a reaction that's typically occurring. Alpha ketoglutarate. The structure here is some amino acid that has an extra amino group on it. Our pyridoxal phosphate and whatever aminotransferase is going to catalyze this reaction. So we go from this L-amino acid to an alpha keto acid, and we go from alpha ketoglutarate to glutamate. Okay? And we could plug glutamate in here and get glutamine as well. And it's really just kind of passing this amino group back and forth to wherever it needs to go. Okay. But what this aminotransferase allows us to do is to safely transport ammonia out of the bloodstream. Okay, so we're getting rid of an ammonia group, an amino group in there.
What transferase?
Aminotransferase. Yeah. Yep. So that's going to be our. Oops. That's going to be our theme. Oops. Come on back here. Transaminase or aminotransferase. Same thing there. All right. But that's going to be our mechanism for moving nitrogen so that we're not making ammonia in the bloodstream. Okay. So glutamine, temporary storage in there, but it gets processed in the intestines, the kidneys, and the liver. And we can see basically our glutamine synthetase. We can actually make this by using ATP to add an amino group here. It requires energy to do that. Our glutamyl phosphate synthetase, yeah. So there's our second step in here. We then will, so we activate the carboxylic acid with a phosphate group, and then we can replace the phosphate group with an amino group, and then we can send this to the liver to remove that. Right. So this, sounds a little inefficient. It's like, oh, we're gonna spend ATP to put this amino group on there, and then we go take that amino group off. All right. But this is how we get rid of excess ammonia in our outer organs, and we can send it into the liver to start processing stuff for the urea excretion. I feel like I'm saying too much. It's this front moving through here. Okay. All right, so let's take a look at pyridoxal phosphate, and then of course pyridoxamine phosphate as well too. So this is an intermediate. It's often enzyme bound as a carrier of amino groups in there. Our aldehyde can react reversibly with amino groups. Here we have our aminated form. Here we have our aldehyde form. So we basically utilize this carbonyl carbon here as our target to add an amino group, and then we can kind of subtract this off later. But this is our pyridoxal phosphate, our lovely little cofactor. And again, we could look at the electron withdrawing properties of the phosphate group up here to allow this carbonyl carbon to be the active site and so on and so forth. But that's organic chemistry, just kind of a reminder of how this kind of grows out and becomes applied. So is this a good structure to know? I don't think I necessarily want you to memorize the structure, but I definitely want you to know about pyridoxal phosphate and what it does and how we're essentially passing an amino group onto the pyridoxal phosphate, and then that will get transaminated onto a keto acid, L-keto acid, or glutamate. And that this is a reversible reaction. So we're constantly adding this and then oxidizing. Pyridoxal phosphates covalently linked to the enzyme and the resting enzyme. So if we look at our transaminase, we're going to see our pyridoxal phosphate will be bound here. All right. We make an internal aldamine in here, and we end up with a shift base. Okay, so you can see here, we look at this. Here's our enzyme. We have a lysine out there. Our group for our lysine has that amino group at the end, and that can interact with pyridoxal phosphate to make this lovely little shift base. And now we have our pyridoxal phosphate covalently bound to our protein. Okay. We can kind of look at the active site in here. This is just an image in case you'd like the images in here, but you can see the pyridoxal phosphate inside this active site cleft. Okay. So pyridoxal phosphate is involved in multiple reactions, all right? We primarily talk about, transaminations, but we can also look at racemization of amino acids, and we can also look at decarboxylation of amino acids as well too. So that's why we have this A pathway in yellow, this B pathway in green, and our next slide we're going to see, or in gray, and our next slide we're going to see a C pathway in green. This is also one of these slides where there's a whole bunch of text at the bottom that kind of goes through that. This goes into really good detail about what's happening here. Here is our pyridoxal phosphate that is bound in here. And we have our L-amino acid, so we can actually kind of get this to start interacting with our pyridoxal phosphate. So now we have an external aldimine where the pyridoxal phosphate is no longer bound to the enzyme, but it's bound to the substrate. And then we go on to form this quinonoid intermediate, so on and so forth. We can transfer that amino group to the keto acid in there, and we end up regenerating our pyridoxamine phosphate. All right. So we've now kind of accepted that amino group from our substrate, and we formed a keto acid in there. Okay. There's another pathway from this shift-based intermediate. We can actually do this racemization where we can kind of change things around. So you notice that we've now gone from an L-amino acid to a D-amino acid. Okay. That is a lesser pathway, and we're not going to be too concerned with that. We can also look at decarboxylation in here as well too. Again, our key factor with our pyridoxal phosphate is we make these quinonoid intermediates in here, we can our pyridoxal phosphate will go back to form that internal aldimine in there, and we've essentially decarboxylated the amino acid that was out here by removing that carboxylic acid group. It gets released as CO2. Okay. So there's a decarboxylation reaction as well too. So all three of those pathways are important. We're going to focus on the deamination aspect though, but decarboxylation, can help us regenerate. Oftentimes we'll see, multiple arrows indicating multiple steps in the pathway. Here we go. This is one of those ways we can get that decarboxylation going. All right, but anyway, let's just keep our focus back on transamination and moving around the amino groups. Oxidative deamination occurs in the mitochondrial matrix. So, I always kind of laugh as I ended up studying the mitochondrial proteins in particular, but hey, everything happens in mitochondria. It's the most important organelle. Okay. Not necessarily true. But we can use either NADH or NADPH in there. So we've got a couple electron acceptors in there. Ammonia gets processed into urea. So we end up, here we have this nice lovely little amino group in here. So for the ammonia excretion, we want to have this trans deamination, which is basically a combination of a transamination and then an oxidative deamination. So there's a kind of two-step process in here. We'll often just draw the arrow from glutamate to alpha ketoglutarate, but really we're kind of doing two things to get this going here. All right. You can see our electron transfer where we make this lovely little aldimine in here, and then we hydrolyze over top of that to regenerate our carbonyl carbon. Actually, to make our carbonyl carbon and get our alpha ketoglutarate and do the transaminase portion, the deaminating of our transaminate. Okay, so we can also look at this in turn. We've sort of seen the glucose alanine cycle in the muscle before. Our liver, we send the glucose to the muscle. The muscle uses the glucose for energy. Ooh, look at me flex. Okay, not really. Should say when I split firewood and then get sore, make a lot of lactic acid in here. But anyway, our glucose, gets converted into pyruvate as we have the energy to power the muscles in here. Our pyruvate, we can then see this glutamate. We can do a transamination reaction here, this alanine aminotransferase. We're going to transfer the amino group from glutamate to pyruvate to make alanine. And we make alpha ketoglutarate within the muscle cell, okay? But the alanine can then get transported back to the liver, where we see another alanine aminotransferase that's gonna do basically the reverse reaction. We're gonna make glutamate, make more pyruvate, which can then go back into gluconeogenesis to make glucose and send it back there. So here's our glucose alanine cycle. And what we've done is we've essentially removed an amino group from the muscle and taken it to the liver, where we can then send that amino group via glutamate into the urea cycle to excrete that excess ammonia. Okay. All right. And again, just to reiterate, why do we need to get rid of pyruvate? Because if we don't, we'll convert it into lactic acid, get that excess lactic acid build up, and you feel the burn in there. I took my kids to see the ACC Hockey League championship, some of the games this weekend, and my daughter was asking, why do they get on and off the ice so much? And I'm saying, well, your typical hockey shift is around 45 seconds because they're skating so hard, they're using a lot of energy in those leg muscles, and they'll build up lactic acid. So then they need like a minute or two to kind of rest that. And if they're out there, longer than a minute or so, then you'll see them get really slow as they start to feel a burn from that buildup of lactic acid in their muscles. All right. Yeah. So excess glutamate then gets metabolized in the mitochondria of hepatocytes. All right. So we can take our glutamate, and oh dear Lord, now we're getting into the crazy figures, right? So where does the glutamate come from? We can actually have it directly imported in there, or we can have glutamine gets transported in there, and then a glutaminase will deaminate that. So here we have our ammonia to make us glutamate. And we can also see oxaloacetate being converted into aspartate to make give us some here. Our glutamate can make the alpha ketoglutarate and donate the amino group to oxaloacetate to make aspartate, and then aspartate can be exported out of the mitochondria. But central to this, and here we're kind of recycling ammonia, okay, because it goes from the glutamate to the aspartate and then outside there. But we can also, just metabolize the ammonia and send that in here, either from glutamine or glutamate. We have this ammonia molecule that is going to be acted on by carbamoyl phosphate synthetase 1, all right? And we're going to generate carbamoyl phosphate here in the mitochondria. This is going to actually be in a combination of ammonia and bicarbonate. It's going to require 2 ATP to do this. And we end up making this lovely little teeny tiny molecule in here. So here's our HCO2. We have our NH4. Basically, we have a bunch of protons that end up in here. And here we have a phosphate group. Where's our other phosphate group? That's actually used to power it. So we're using one to put it on here and one to charge the interaction with our carbamoyl phosphate synthetase. Okay. We'll then see carbamoyl phosphate can interact with ornithine to produce citrulline in the matrix. And then this is going to be exported out of the mitochondria, and ornithine gets imported in for this reaction to occur. But carbamoyl phosphate and CPS1 are our key molecules and enzymes for our transaminase to produce urea for elimination. Okay. All right. So ammonia is recaptured through the synthesis of carbamoyl phosphate. Okay, this is the first nitrogen acquiring reaction of the urea cycle. So obviously you're going to see this is going to be a target of regulation, right? Woo, yay. Okay. So the first thing we do is we're going to charge this bicarbonate with our ATP in here. We're going to add a phosphate group on there. So now we have an activated carbon, and this activated carbon will then allow us to attach the ammonia group on there and kick off the inorganic phosphate. All right. And then once we have made our carbamate, we can now charge this again and put another phosphate group on the carbon. So we make our carbamoyl phosphate, right? So bicarbonate to this carbanic phosphoric acid anhydride, add the ammonia to get the carbamate, and then phosphorylate that again to get carbamoyl phosphate. Using up 2 ATP, you can also bet this is going to be highly regulated because we are using a lot of energy in here. But that's essential. That should also kind of tell you how important maintaining nitrogen balance is as well too. You don't want to die in a soup of your own ammonia. All right. The nitrogen from the carbamoyl phosphate enters the urea cycle. Okay. So here we have our carbamoyl phosphate. I know this picture is maybe a little hard to see here. Blow this up and separate this out into individual components coming up here. But we basically combine the carbamoyl phosphate with citrulline. Okay, so sorry, ornithine to make citrulline. So ornithine is one of our amino acids that does not get incorporated into proteins, but our ornithine reacts with carbamoyl phosphate to give us citrulline. And here you can see we have, our nitrogen group on here that's been attached. So our ornithine 1, 2, 3 carbon, 5 carbon in here. And now we have added a 6th carbon with the carbamoyl phosphate, but we've also added another amino group. We can then transport the citrulline out of the mitochondria, and we see our arginosuccinate synthetase in here. And you're like, oh, okay, that's our connection with the citric acid cycle, right? So we will make the citral AMP intermediate, right? Something crazy looking like this, which is why I don't really, want you guys to memorize a lot of structures. It's probably a good idea to know the carbamoyl phosphate structure.
Can you say that?
Yeah, I don't think I'm going to ask you for the structure of carbamoyl phosphate, but I'll check. I'll update my study guides if I do have a question about the structure of carbamoyl phosphate. But I think that's helpful so you can sort of see where it comes from, and that's why I like pictures, slides like this. Even though this is a very busy slide, we kind of color code where everything is coming from here. So you can see here's our ammonia that came from our transaminase. Our carbon comes from the bicarbonate, and then here we have our ornithine, whatever in here. Here is our citral AMP intermediate. All right, we're going to see aspartate interact with this. Okay, and we have another ammonia group here, and now we've added basically this arginosuccinate in here. All right, so arginosuccinase is going to cleave off the carbons that came from aspartate that are part of the arginosuccinate to give us fumarate. So again, we're in the citric acid cycle, and we're going to produce arginine in this case as well too. And then our arginase will deaminate the arginine to give us ornithine. And here we have our urea molecule. So we're essentially doing all this work over here on the right-hand side of the slide so that we can kind of set up our urea in the center of this big molecule. And then first we cleave off the fumarate, and then we can cleave off the urea. So let's see here. We're using up ATP, 2, 3 ATPs in here. Yeah. All right. And it's kind of neat because this third ATP, the one that we use outside the mitochondria and the cytosol here, we're actually aminating. So instead of adding a phosphate group like the first two do, here we're actually adding the adenosine group, adenosine monophosphate group in there, so that we can condense that with aspartate to give us this arginosuccinate. Okay. All right. But then again, once we make our urea, ornithine can get transported back into the mitochondria, and we are good from there. Okay. Oops. Do that. Okay. All right. This is basically just a bigger picture of that without all the words on the side there. But this should allow you to kind of follow the reactions that are going in here. We notice that we call this citrulline to making arginosuccinate. We have a 2A and a 2B, where we have this kind of intermediate, this big charging intermediate in here that because we have to add the adenosine monophosphate to the citrulline so that we can interact it with the aspartate so that we can get this lovely little arginosuccinate. And you see we're releasing our adenosine monophosphate here. So 2A, 2B, it's kind of like a two-step process. Really, we'd probably draw this citrulline to arginosuccinate, but this just shows you how that's happening. Okay? And again, I like the color coding. We're just going to cleave off this little 4-carbon unit here to make fumarate. All right, we'll feed that back into the citric acid cycle. We're left with arginine. We cleave off the ends of arginine here, and we make aspartate and leave behind ornithine. Okay. Good stuff, right? I told you this chapter is where it starts to go crazy. Yeah. Which is why I'm going to find a second here in a second to talk about something different, because I know this is a lot. This is where, you know, you start looking at this, and if you don't develop a way to kind of look at what's going on here and kind of break down each of these pathways in a way that, oh, okay, I see what's going on. You know, you don't have to memorize this pathway, but you should be able to look at this and say, okay, so here's where this reaction is going. Here's where this is coming from. Here's where this is happening. And then, of course, later on, we're going to talk about the regulation of these enzymes. Woo! So, yeah, there's just so much that goes on in here. Okay. Yeah. That's what I felt like yesterday. Yeah. I was actually very kind. My wife was studying for a certification exam, and she was over the weekend. So I'm like, well, I'll take the kids to the hockey games for 4 hours. So my daughter is exhausting because she talks incessantly, like literally. She's like, okay, I'll be quiet. Yeah. You know, it's funny because I talk a lot now, but my entire family is very, very quiet. So my wife is always like, well, you said you talk a lot. I'm like, yes, but your Indonesian family talks a lot and is loud. And that's the thing. I talk a lot, but I'm quiet. I'm soft-spoken. I'm not shrill. Her voice just grates on me. But yeah, it's very interesting. Yeah. Well, and it's just, you know, comparing kids. I could bring my son to class and he would sit there quietly and entertain himself, or read or play iPad or something. Just, you know, I had to look over to make sure he was still there, you know. And yeah. When I would bring my daughter to class, the same age I brought my son, actually a little bit older, she would be up in the front of the room. It's like, why are they asking questions? You know, they literally could not go more than 3 or 4 minutes without coming up and interrupting. So yeah. And she has to be the center of attention. So totally not like me. Yeah. All right. So, you know, when aspartate gets into the urea cycle, this is basically our second nitrogen acquiring reaction. So the first one is the formation of that carbamoyl phosphate. We've got one amino group on there. When aspartate comes in, we get our second amino group, right? And that's going to give us that arginosuccinate, where we cleave off the fumarate, and then we can cleave off the urea that has 2 amino groups in there. Right. Like my hand gestures in here. I don't think so. Yeah, they're it's there somewhere, but it might be nitrogen excretion or nitrogen fixate, nitrogen balance, maybe. But the urea cycle is, yeah, it's not like the Krebs cycle or the TCA cycle or the citric acid cycle in there. It's pretty much just that. All right. So once we've made our arginine, okay, we've cleaved off our fumarate, we have our arginine, we can then cleave off the urea through this arginase enzyme, and then we regenerate ornithine in there, okay? And of course, if we have arginine from some other system, that can just kind of feed in here as well too. So much like we saw the citric acid cycle and all those anaporotic interactions, arginine can come in at some point. Arginosuccinate, same, not that we have as much of that. But anyway, we regenerate ornithine, we can transport it back into the mitochondria, and it can go through another round in there. Okay. So aspartate arginosuccinate, there is a shunt that links the citric acid cycle to the urea cycle. So we see our citric acid cycle occurring in the mitochondria, right? Ooh, yay, okay. Part of our urea cycle is in the mitochondria, really just kind of the initial step, and then everything else happens in the cytosol. But in our citric acid cycle, when we have oxaloacetate, we can generate aspartate from that, which can get transported out, which can, of course, then interact with our citrulline to give us our arginosuccinate, really our citrulline AMP, intermediate that's in there. That's why I had that parenthetical part. But you see, we don't draw that in here as well. We're just saying citrulline and aspartate are going to interact to give us that arginosuccinate. And then we get our arginine, but we also get fumarate. And then fumarate can get converted to malate and back into the mitochondria. You can see fumarate to malate in the mitochondria and fumarate to malate in the cytosol as well too. We can also see here making oxaloacetate and malate to again put this back in here. We see our electron donors, NADH in there. And so on the outside, we use NADH. On the inside, we regenerate NADH as we go from oxaloacetate to malate or malate to oxaloacetate. Right. Same way here, alpha ketoglutarate to glutamate in the cytosol, glutamate to alpha ketoglutarate in the mitochondria. And we're basically transferring that amino group from glutamate to alpha ketoglutarate. And here we are aminating the alpha ketoglutarate to make glutamate. So if you think back to like chapter 4 or 3, where you had to memorize all the structures of the amino acids, awesome. This is why it's helpful, because now you can say, oh, really? Well, I know the structure of glutamate. If I just take away that amino group that's on there, I get alpha ketoglutarate. Or if I add one to the R group, I get glutamine. So I think that's why some memorization is helpful, because instead of just having all these random terms, like what's the structure of this one, what's the structure of this one, you're like, oh, we're just varying by one group. We're transferring one group. And in this case, for this chapter, we're transferring amino groups over and over and over and over again. All right. So let me pause and look at my slide number here. Okay. So thinking, let me look ahead real quick here. Yeah, okay, we'll do a little bit more and then we'll, so let's talk about the regulation of the amino acid cycle. I think this does tie in nicely. I don't want to overwhelm in here, but since we're talking about CPS1, and you'd say, that means there's probably a CPS2, right? Yeah, there is. But CPS1 is regulated in a variety of ways. This is a very important cycle in here. So we're going to see a lot of regulation. It gets activated by the presence of N-acetylglutamate. All right. So N-acetylglutamate is formed by N-acetylglutamate synthase, obviously. But basically, when we have high glutamate concentrations and high acetyl-CoA concentrations, that's when we're going to see our N-acetylglutamate production. I'm going to say even when we have high glutatamate, too many typos. And our N-acetylglutamate synthase is activated by arginine, right? So here you can see we have two amino acids. If we have high concentrations of them, we're going to end up, so these are kind of these proteins, or these amino acids kind of function as our protein sensor, or in a way, our nitrogen sensor in there. We've got lots of protein coming into the body. We're going to need a way to get rid of that, okay? We will see that the urea cycle enzymes are responsive to changes within the body. So if you eat a high protein diet, if I have like a bag of beef jerky every day, oh, that sounds delicious. But I'm going to see a lot more of these enzymes being produced in here. We also see urea cycle enzymes increase during starvation. Why? Because you're starting to break down the proteins of the body for energy needs, because you don't have enough carbohydrate or lipid sources of the energy in there. So it's kind of counterintuitive. Oh, I'm eating a lot of protein, or oh, I'm not eating much of anything at all. So it just depends whether you have dietary amino acids or recycling amino acids. Recycling cannibalization, basically. Okay. So here's what we see. Our glutamate and acetyl-CoA can be combined to make this N-acetyl-glutamate. This enzyme is upregulated by the presence of arginine. N-acetyl-glutamate upregulates CPS1. Right? All right. So lots of protein. We're going to see increased activity of carbamoyl phosphate synthetase. All right. We talk about essential amino acids versus non-essential, and then I love this, conditionally essential amino acids. All right. So our essential amino acids are ones that we cannot generate ourselves. We have to get them from our diet. Okay. And these include histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. Okay. This may fall under, this chart might be a good thing. I have to look at my questions. Let's see how I ask my questions here. Which of the following is essential, or which of the following not? But our non-essential alanine, asparagine, aspartate, glutamate, and serine. It's probably easier to memorize the non-essential ones. It's like AAAGS. All right, there you go. All right. Conditionally essential are the ones that they're required to some degree in growing animals that are during illness. You may not be able to get enough of them at certain stages, right? It's weird for me. I never really heard of growing pains, but I can remember when my son was going through a growth spurt, his legs would hurt because just this really physical growing. I thought it was just like the name of a cheesy '80s TV show with Alan Thicke. Man, I'm old. But it's an actual thing, and that may be dependent on these conditionally essential stuff there that we're trying to grow, and we need more of these proteins in here. All right. But the idea is, from a well-balanced diet, a variety of foods will supply all of this. So, don't just eat chicken nuggets every day. Yes, actually, that would give you all your protein and probably a good amount of fat and salt. Anyway, if we look at the amino acid degradation of various proteins, this is also another fun part here too. Like, where do these? Yeah, I know you guys are like, we're gonna hate this chapter. This is gonna be a chapter where everybody's like, "Oh, I'm sorry, I signed up for this class. Too late to drop." No. Yeah. If we look at our central metabolic pathway, here we see our intermediates in here. Some amino acids can give you multiple intermediates, more than one in there. Ketogenic amino acids can be converted to ketone bodies. We can get 7 of them that can make acetyl-CoA. So we see if we have excess acetyl-CoA, we can get those ketone bodies. So leucine, isoleucine, threonine, lysine, phenylalanine, tyrosine, tryptophan are ketogenic. The ones in red are also glucogenic. All right. 6 of them can go to pyruvate, 5 of them alpha ketoglutarate, 4 to succinyl-CoA, 2 to fumarate, 2 to oxaloacetate. And let's see, do we have any duplicating? No. All right. This is also why, and this is one of the things I never noticed, but diabetes is not something that runs in my family, but I would every now and then see like food containers that would talk about their glycogenic index and things that might have been, say, high in tryptophan. All right. And you have to say, oh, okay, this has got tryptophan, so it has a glucogenic index number in there based on the quantity of tryptophan or threonine or whatever in there. And these are glucogenic because they can feed into our glycolytic pathways and ketogenic to make our ketone bodies. Okay. For me, I look at this and I just go, well, okay, gonna memorize this. Ones in red are different. So I like this slide. Other people like this presented this way a little bit better. Again, here we do color code a little bit. We can see our ketogenic ones are in blue and our glucogenic ones are in orange, red, pink, I don't know, whatever color that is. But again, we can see where they feed in. Yeah. Well, it's not really red. It looks kind of like peach to me, an orangish peach color. Yeah. But I'm never sure if it's just like the screen color or because there are things too. Actually, these projectors are really good, but I know in the old building I'd project stuff and I was like, that is a completely different color than what I have on my laptop screen here. So, But yes, so you can see kind of where these feed in. And again, we can recall where we get our ketone bodies from that we talked about with our excess acetyl-CoA feeding into our citric acid cycle. There we go. And of course, from oxaloacetate, we can feed into the gluconeogenesis to make glucose. Me, I'm boring myself. No. Nope, better cut that out of there. All right. If we look at the way these degrade, you can see all sorts of our intermediates in here. I am not going to ask you to know this in any great detail. I just want you to be aware that these pathways do exist. I mean, I'm not going to ask you about alpha keto adipate or glutaryl-CoA or anything crazy like that. But on the take-home portion, you might get, say, tryptophan breaks down into blah, blah, in this disease state, blah, blah, blah. Why would this be so? And then maybe there's a step in here. Even this, you see there's like 4 steps before you get from glutaryl-CoA to acetoacetyl-CoA. It's, unless you're studying exclusively this particular pathway in minute detail, we can gloss over it at this level, right? The important thing is just to see that, all these things somehow in the boxes, these are our ketogenic ones that are breaking down, and you can see the pathway of whether they make acetoacetyl-CoA, acetyl-CoA, or succinyl-CoA. Right. The interesting thing is, as we break these down, our intermediate metabolites can be used to synthesize other biomolecules as well too. So for example, tryptophan, we can see here, I could, hold on a minute, come on, there we go. I could take my tryptophan and I could convert that into acetoacetyl-CoA through a bunch of steps in here, make pyruvate and alanine or whatever in there. Or I could use the skeleton to make nicotinate, niacin basically in there. And if I want my niacin adenosine diphosphate, I can make my NADP building off of the skeleton. Or tryptophan, if I cleave off this side group in here and hydroxylate this, actually, I guess I'm not really cleaving it off. Am I just cleaving off something? I can make serotonin. Woo, yay, okay. Or we can look at this indole acetate, which is a growth factor for plants. But tryptophan has multiple fates in here, okay? So I don't need to carve everything up into its individual components. It's kind of like our Legos. This would be, tryptophan would be one of our little block sections that can be used to build a whole bunch of stuff. All right. Wait till we get to the point where, well, how do you make tryptophan de novo? Okay. Oh, yeah. Probably don't have to memorize it. Oh. Yeah. All right. Just like we talked about with fatty acid oxidation diseases, there are a lot of diseases for protein degradation as well too. Genetic defects and the many steps of phenylalanine degradation can lead to disease, okay? So where is it? PKU is the one here. So the first step is, degrading phenylalanine into tyrosine in here. This requires phenylalanine hydroxylase. If we don't have phenylalanine hydroxylase, or if there's a defect in it, you get phenylketonuria, which is that the maple syrup urine disease? Actually, I don't think so. I think it's slightly different. But there's a variety of these diseases, tyrosine 1, 2, 3, and this alkaptonuria. But you can basically see every enzyme involved in here. If there's a deficiency in it, there's a disease for it. Maybe not malonyl acetate isomerase. Apparently that one's fine. We don't really need that one. No, I'm just kidding. I'm sure if you accumulated that, you'd have another disease in there too. But so phenylketonuria is that first step in there. It impairs neurological development and leads to intellectual deficit. And it's one of these things where, well, how do you fix this? And the simple answer is, you go to the doctor and says, it hurts when I do this. And the doctor says, well, don't do that. Well, for phenylalanine, that basically control the diet so that you don't eat a lot of phenylalanine. Now, we do need phenylalanine, so you have to have very small amounts of it in there, but you just want to make sure there's not an excess of phenylalanine, or else you'll get that buildup in there, and that neurological deficit can be pretty devastating. But if it's well controlled, so again, you often see a lot of food products will list their phenylalanine content as well too. All right. I need to go find some. But I've seen phenylalanine listed as an ingredient on some of those. Because phenylketonuria it's rare, but it's not super rare. It might be like 1 in 10,000 people will have phenylketonuria. So yeah, it's enough that there's probably several dozen here in the metro area with it. Then people have to watch their phenylalanine content. We can also look at various amino acids being degraded into pyruvate. We have our alanine, again, tryptophan in there, multiple steps we can make alanine, pyruvate. And you can see, hey, we're actually making glutamate. So as we degrade tryptophan to pyruvate, we make another amino acid. So it's not like we're just we're shoveling stuff around from here to there. Again, you see in a lot of these pathways, pyridoxal phosphate is involved as a cofactor. Folate, we kind of talked about. I think one of these had biotin in there as well too. Yeah, biopterin. Yeah, yep. So we have our single carbon transfer that's involved in there, and you can see what we cleaving off here. Yeah, I love this the glycine cleavage enzyme. We just kind of ran out of names for them. If we look at glycine, because it's such a small molecule, we have a couple different ways we can degrade it. We can hydroxylate it to serine and then convert serine to pyruvate. We can use this glycine cleavage enzyme, which is most common in us, where we separate the 3 central atoms in here. So we're basically cutting all those carbons out in there, and we'll release carbon dioxide and ammonia, again, to go into urea cycle. And then the methylene group will actually get transferred to the tetrahydrofolate on there. We also have a D-amino oxidase in here, which is a minor pathway. Keep in mind our amino acid, this glycine is the non-chiral one, so this D-amino oxidase would recognize it and as a substrate in there. And it'll eventually make oxaloacetate. But this third pathway is a major component for the formation of kidney stones as well too. So, all right. We look at alpha ketoglutarate. I'm going farther than I wanted, aren't I? All right. We'll do alpha ketoglutarate and then we'll stop there for a moment. So arginine to ornithine. So when our amino acids go to alpha ketoglutarate, we can all convert, we see a bunch that get converted to glutamate, particularly our arginine and glutamine, our nitrogen containing ones. Our positive side chain amino acids, and then proline and histidine that have those ring structures. All of those can get converted to glutamate, and then we can deaminate glutamate to alpha ketoglutarate in there, and that deamination feeds into the urea cycle. So the arginine degradation is part of the urea cycle. So we actually get our ornithine that feeds into that. Recall from like a half hour ago. Okay. So let me pause here because I know this is a lot. We'll talk about the branch chain amino acids a little bit on Thursday and finish out this chapter. But there's something else I want to do on Thursday as well too. So let me stop sharing. What's that? Yes, the case study. Yes. So if I pull up the, all right, I got to find where I put this. I think it's in assignments. Well, there's urea cycle case study here. Okay, this one. Yeah, let me check and make sure this is the right one. Maybe. Come on. Show me the case study. What's that? Okay, yeah. See, that for me, I was already old when that reference
Hey, Dr.
Sorry
Eager.
about that. Yeah, I ran out of battery. So I had too many things going there. But we're essentially done for today. Just check the announcements. There's going to be a case study posted. So we'll let you guys go because we're about out of time anyway. But we'll pick up with this on Thursday. So
Okay, thanks. I'll see you tomorrow.
yep. All right. See ya. Sorry for that abrupt end.
Oh no, it's fine. Yeah, I figured something bad must have happened when you just like disappeared. So
Yeah. Just, all right. Catch you later. Bye-bye.
you too.